welcome to our 9th lecture on high temperature stress. This module is going to be divided into two parts. The first part is going to be about plant responses to heat, and the second part is going to be about plant resistance to heat stress. In this part of the module, we will go over the following: organisms' classifications according to their thermotolerance, injury, factors affecting injury, measurements for injury, and types of injury. Afterwards, we'll go over chronic injury and define its characteristics, see how that affects CO2 fixation, Rubisco activation, and then we'll have a comparison between the C3 and the C4 plants in chronic injury for heat stress. And finally, we will go over acute injury, where we will talk about its characteristics, its effects on lipid membrane, and its effects on proteins. We have now reached the portion of the course where we will apply all the information and principles from the previous sections to plant responses to temperature extremes. Fortunately, most plants do not experience the extreme temperatures indicated by this photo of a forest fire on Montana's Bitterroot Valley in 2000. However, some pines require forest fires to release seeds from serotonin cones. The high temperatures melt a resinous coating, paving the way for seeds dispersal. In some other species, smoke can promote seed germination. Our primary focus will be on vegetative portions of the life cycle rather than resistant structures like seeds. Temperature ranges triggering heat stress injury can vary tremendously among organisms. Accordingly, organisms can be classified according to their thermotolerance. Psychrophiles are cold-loving and subject to heat damage above 35 degrees Celsius. Examples include many algae which live in thermally buffered environment. Mesophiles are damaged at 35 to 45 degrees Celsius. Examples include higher aquatic and shade plants. The third group are the thermophiles, which comprises a broad group that is subdivided into moderate and extreme. Moderate thermophiles can be damaged at 45 to 65 degrees Celsius. Since this group includes many higher land plants, it is an economically important group. Extreme thermophiles are remarkable organisms. They are primarily in the archaea domain and can be found in extraordinarily hot environments. Those can go up from 70 to 80 degrees Celsius. There is no single universal mechanism of injury at higher temperatures. Modes and severity of injury can vary based on factors such as length of exposure, prior environmental conditions, plant species, tissue, and stage of development. The multiplicity of injury mechanisms presents some challenges in measuring plant responses to heat stress. The most basic responses is whether or not the plant survived the stress. However, sublethal doses can affect many plant processes. Therefore, it is not surprising that many indices are available to detect damage to specific cellular components or processes. Examples include chlorophyll fluorescence to monitor photosynthetic efficiency, monitoring breakdown products of oxidative reactions, including low molecular weight hydrocarbons and aldehydes from lipid peroxidation, and using electrolyte leakage to track structural and functional integrity of the membrane. Electrolyte leakage is a simple procedure that can be used to measure loss of the ability to maintain concentration gradients, either through impaired transport systems or structural damage to the membrane. Plant tissues can be placed in distilled water following heat stress. The conductivity of water to electricity is low. However, adding electrolytes increases the conductivity, which can be measured using a probe with suitable electrodes. If the plant cell membranes are damaged, electrolytes will leak out and increase the conductivity of the solution. Responses are usually presented as a percentage of total conductivity. The plot on the right shows a typical relationship between electrolyte leakage versus exposure temperature in Celsius for a moderate thermophile. The midpoint Tmid of the sigmoidal response curve is frequently reported in the literature. The time-temperature interaction is significant for several reasons. Scientists like to make tables comparing heat tolerance of different species, but these comparisons are of limited value unless the experiments were conducted similarly with comparable exposure durations. However, experiments are frequently conducted with conveniently short durations, 15 to 30 minutes. In reality, plants out of doors are exposed to high temperature for extended durations, with critical temperatures being correspondingly lower than short duration studies. It is also important to recognize that different mechanisms of injury may be involved in damage at short and long durations. Since there can be different mechanisms of injury involved in short and long durations, scientists usually distinguish between acute and chronic exposure. Our next slide set will deal with chronic heat stress injury. Chronic or indirect injury is characterized by exposure to moderate temperatures for long time periods, usually days to weeks. Chronic injury is normally associated with metabolic lesions. Our first example deals with relative reaction rates within a reaction pathway. Consider a reaction pathway in which enzymes sequentially catalyze conversion of substrates to products. Many factors affect the reaction rates, including concentrations of substrates at less than saturation, concentration of catalyst, and temperature. It is well accepted that enzymes increase reaction rates by lowering the free energy of the transition state. Here we look at the temperature effect on enzyme activity. As temperature goes up, molecules have more energy and collision rate increases, which leads to an increase in the reaction rate. Temperature effects on reaction rates apply to both catalyzed and uncatalyzed reactions. Once temperatures become very high, enzymes start to unfold and protein denatures, resulting in enzymes' loss of function, which in turn decreases the rate of the reaction. If we come back to our example of a reaction pathway, there will be several sequential reactions that are affected by temperature. Simplifying the situation by making some assumptions on reaction order and enzyme concentration, the rates of the reactions can be described by rate equations involving rate coefficients K and concentrations of substrates. Virtually all reactions will speed up with temperature increase in the moderate range. However, different reactions have different magnitudes of free energy of activation. So an increase in temperature can either speed up or slow down different reactions to a different extent. If such change would happen and affect enzyme 1 in our example, it will indirectly affect enzyme 2 and all dependent reactions beyond that point. The crossover from a balanced to an unbalanced situation can lead to a metabolic lesion involving either a shortage of essential metabolite or the overproduction of a compound to the point of toxicity. In addition to imbalances within a reaction pathway, chronic heat stress can also lead to imbalances between metabolic pathways. For our example, we will use respiration and photosynthesis. Differential high temperature inactivation of these related pathways can lead to growth inhibition and starvation. Both photosynthesis and respiration increase with increasing temperature until maxima are reached. Then rates decline at higher temperatures. However, the temperature optima are different for the two processes, with photosynthesis being more heat susceptible than respiration. To explore the temperature sensitivity of photosynthesis, we can start with the Calvin cycle, which is more susceptible to heat than light reactions. Rubisco is the enzyme that triggers CO2 fixation in the Calvin cycle. The reduction of CO2 stores energy in chemical bonds and creates raw materials. Since chloroplasts contain both carbohydrate synthesizing and degrading enzymes, steps must be taken to prevent futile cycles. This is achieved in part by having synthesis on and degradation off during light periods. To control synthesis, Rubisco has active and inactive states. The inactive state is when sugar phosphates are attached to its active site, and the active state of Rubisco is when sugar phosphates are released, carbonylated added COO, and Rubisco binds magnesium. The process is triggered by the enzyme Rubisco activase, which uses ATP hydrolysis to activate Rubisco. Rubisco activation may be the primary heat labile step in photosynthesis in some plants. The figure shows differential thermostability of Rubisco and Rubisco activase, with activase activity dropping off at about 44 degrees Celsius, and Rubisco being stable to at least 60 degrees Celsius. At least partially due to the thermosensitivity of the carbon cycle and Rubisco activase in particular, photosynthesis has a lower temperature optimum than respiration. The difference is approximately 20 degrees Celsius for potato leaves. The consequence to the plant is that at moderate temperatures, carbon is reduced at a greater rate than it is oxidized. At high temperatures, carbon is oxidized faster than it is reduced. There is a critical temperature called temperature compensation point, or TCP, at which the respiration rate equals the photosynthetic rate and the plant breaks even. At higher temperatures, there is a net loss of energy leading to starvation over time. This represents an example of growth inhibition or starvation from differential high temperature inactivation of related metabolic pathways during chronic heat stress injury. Differences have been observed in how C3 and C4 plants are affected by chronic heat stress injury. As it turns out, C4 plants have higher temperature compensation points than C3 plants. The higher TCP in C4 plants relates to their ability to reduce or eliminate photorespiration. Photorespiration is a problem in C3 plants because Rubisco can act as an oxygenase as well as a carboxylase. When photosynthesis was invented, there was essentially no oxygen and more CO2 than there is on Earth today. It appears that photosynthesis never addressed the oxygenase activity of Rubisco directly, at least in C3 plants. Photorespiration consumes O2 and evolves CO2 in light, wasting Rubisco and ATP. The glycolate formed is nearly useless and is oxidized in the peroxisome to recycle carbon, generating H2O2 in the process. Overall, it reduces the photosynthetic efficiency. C4 plants have a way to deal with this problem that includes a different anatomy and the fixation of CO2 twice. The first fixation occurs in the mesophyll cells. CO2, when it's in the more soluble form of HCO3, adds to phosphonol pyruvate PAP, forming a C4 compound, oxaloacetate. Fortunately, the enzyme involved PAP, a carboxylase, does not have an oxygenase activity. The C4 compound is reduced to malate and transported into the bundle sheet cell. CO2 is released, generating pyruvate. In the second fixation, CO2 goes through a normal Calvin cycle. Pyruvate is transported through the mesophyll and converted to PAP, along with ATP being converted to AMP and pyrophosphate. Therefore, C4 plants effectively pump CO2 to Rubisco by dropping CO2 off in the bundle sheath cells. This favors carboxylation over oxygenation, and the C4 plants exhibit little or no photorespiration, making C4 photosynthesis more efficient than C3 in warmer climates due to the elimination of photorespiration. Although C4 photosynthesis provides an edge in warm climates, the relative efficiency shifts in favor of C3 plants at lower temperatures. This is due to a temperature dependency on photorespiration and C4 plants spending ATP to fix carbon twice. The tendency for Rubisco to act as an oxygenase increases with increasing temperature. The temperature dependency involves changes in relative affinity of Rubisco for oxygen and carbon dioxide, and to the differences in gas solubility between oxygen and carbon dioxide as a function of temperature. If, however, C3 plants are exposed to an atmosphere very low in oxygen, the carboxylase activity of Rubisco will be favored over oxygenase activity, and the C3 pathway loses its temperature sensitivity. The higher temperature optimum for a photosynthesis in C4 plants allows them to maintain a positive carbon balance to higher temperature than C3 plants, and is reflected in a higher temperature compensation point. This is not surprising since many C4 plants evolved in the tropics and are well adapted to high temperatures. Acute or direct heat injury is sometimes referred as heat shock. It is characterized as involving short exposures to a very high temperature. As covered in the introductory section, heat stress injury can involve cellular membranes. Acute membrane injury can result from hyperfluidity or degradation of lipids. Hyperfluidity is at the other end of the scale from low temperature responses when the membrane transitions from liquid crystalline to solid gel states. Just as function is impaired when the membrane is too viscous at low temperature, very high temperature can cause the membrane systems to become too fluid and lose structural integrity. In the previous modules, examples of lipid-based signaling induce enzymatic removal of fatty acids by lipases and their subsequent peroxidation by lipooxygenase. Heat stress can lead to much larger scale in situ peroxidation. Extensive peroxidation of membrane lipids leads to breakdown in membrane structure and function. The occurrence of lipid peroxidation can be observed by the production of several low molecular weight hydrocarbons. An example on some of these hydrocarbons is ethane. Many of the membrane lipid breakdown products, including ethane, can be readily assayed by gas chromatography. The graph on the left from studies of heat stress in pepper leaves shows that membrane damage occurred at temperatures greater than 48 degrees Celsius based on electrolyte leakage. Relative to acute heat stress injury, the primary informational item for the graph on the right is that ethane was produced at temperatures corresponding with increased electrolyte leakage in the graph on the left. Although cause and effect are not established, there is an association between lipid peroxidation and loss of membrane function. The ethylene response curve demonstrates a wound response at sublethal temperatures. Malondialdehyde, or MDA, is another indicator of membrane lipid breakdown. MDA can be measured using a colorimetric assay. This example from turf grass exposed to high temperature stress shows elevated malondialdehyde levels associated with the heat stress treatment. Collectively, ethane and MDA assays indicate that breakdown of membrane lipid is associated with acute heat stress injury in a number of plant species. The other primary mechanism of injury from acute high temperature stress involves proteins. Proteins exposed to very high temperatures can denature, unfolding to the point of irreversibly losing activity. Changes in protein stability can span a continuum from subtle conformational changes all the way to degradation. Therefore, stability can be viewed from several related perspectives. Functional, which means maintenance of a conformation that allows normal function, and structural, which means the maintenance of higher order organization and retention of solubility. Protein stability can also be addressed using a thermodynamic approach, involving the change in free energy to go from conformation to another. The overall delta G involves two terms: enthalpy term, inter and intramolecular interactions that tend to stabilize folded structure, and entropy term, unfolding a protein increases S and therefore decreases free energy. Adding these terms together shows that many proteins are only marginally stable. For example, an increase in temperature can increase the contribution of the entropy term to free energy, outweighing the stabilizing effect of bonding leading to unfolding. What factors interact to determine protein stability? Protein properties, more specifically the intra and intermolecular interactions. Second, the solvent system, and that depends on the solvent and the co-solvents. Third of all, physical properties such as temperature and pH. Most enzymes have a pH optimum that is related to ionization of side groups. Protons are added or removed as pH decreases or increases respectively. Changes in charge affect conformation and activity. Loss of protein stability can be a complex process. The simplest case is a bimolecular transition from native to denatured. In many instances, there are numerous intermediates, some of which are reversible. Since non-functional proteins are rarely completely unfolded, the term denatured is frequently used to describe irreversible loss of activity. Once proteins start to unfold, they expose hydrophobic groups that were tucked into the interior. Since this is an unstable situation in an aqueous solvent, the exposed hydrophobic patches tend to stick together, causing proteins to aggregate. We are all familiar with examples of heat-induced changes in protein structure, leading to unfolding, aggregation, and loss of solubility. Egg whites contain several proteins, including albumin. When heated in the frying pan, they become insoluble aggregations. An analogous process can take place with plant proteins. High temperature can lead to unfolding and exposure of normally buried hydrophobic sites. Hydrophobic patches can aggregate. Initially soluble aggregates can lead to turbidity. At higher temperatures or longer exposure duration, aggregates can become large enough to precipitate. In addition to the loss of catalytic activity, the presence of precipitated proteins is generally considered toxic for the cell. Heat-induced damage to proteins can also be observed through loss of activity. This figure shows residual catalase activity following heat stress. In conclusion, both membranes and proteins are primary sites of damage from acute heat stress injury. In the next slide set, we will examine mechanisms of resistance, including the role of heat shock proteins. At the end of part 1, you should know the answers to the following questions. What are the classes of organisms according to their thermotolerance? What are the factors that affect injury severity? How is injury measured? What are the types of injury and what are the differences between them? How does chronic injury cause indirect metabolic lesions? What is the effect of chronic injury on C3 and C4 plants? Name 2 hydrocarbons that are produced under acute heat stress. And finally, how does acute injury affect both lipid and protein stability?